Welcome to The Critical Edge, the no-nonsense podcast breaking down the latest game-changing evidence in trauma and surgical critical care to give you the edge when things get critical.
Glad to be here for another deep dive.
So, picture this scenario. You're standing at the bedside of a 24-year-old MVC victim.
Okay, setting us up.
It's post-op, day 3 in the SICU. You've, nailed his bilateral femurs. You've packed and ultimately resected a shattered spleen.
Right, major kinetic insult.
Exactly. And you have definitive hemorrhage control. The surgical work is technically flawless, but you look up at the monitor and his heart rate is 135.
Oh.
He's requiring escalating doses of norepi. His white count is 25. His vent pressures are climbing as his lungs fill with fluid, and you are staring at a massively edematous open abdomen that you have absolutely no chance of closing anytime soon.
A classic terrifying presentation.
Right. And here's the kicker. You've cultured everything. Blood, urine, sputum, the peritoneal cavity. They are all sterile.
Not a single bug.
Not a single functioning bacterium in this patient's body. Yet his immune system is behaving exactly as if he is in the throes of overwhelming septic shock.
And he is rapidly descending into multiple organ failure.
Exactly. So today we are undertaking a massive deep dive into the foundational immunology of trauma to explain exactly why that patient is crashing.
It's so vital we cover this.
We're unpacking the exact mechanisms detailed in the landmark literature, specifically looking at the pathways outlined by Brown, Kokanor, and Todd regarding the systemic inflammatory response.
The required reading for anyone managing these patients.
For you, the U.S.-trained trauma and surgical critical care surgeon, this is the invisible battlefield you operate on every single day. Our mission today is to connect the microscopic intracellular signaling pathways directly to those macroscopic clinical disasters you manage at the bedside.
Right, from a single receptor on a macrophage all the way to complex coagulopathy.
Yeah, ARDS, and the chronically critically ill patient who simply refuses to heal.
It really is the defining challenge of modern surgical critical care. Because, as you well know, your surgical technique, your rapid hemorrhage control, your MTP, those save the patient's life in the golden hour.
Absolutely.
But it is the patient's own endogenous immune response to that initial kinetic insult that absolutely dictates their survival in the days, the weeks, and, sometimes the months that follow.
You use the scalpel to stop the bleeding, but the immune system decides the ultimate physiological outcome.
Exactly. We have become incredibly proficient at keeping patients alive through the initial shock phase, which means the bulk of our mortality has shifted to the late phase.
Driven entirely by immunological chaos.
Yeah. And to understand that chaos, we have to start with a fundamental causal question that I think we often just gloss over in our training.
Which is?
Why does the human body mount a massive, systemic, highly destructive immune response to sterile trauma like a closed femur fracture? In the exact same way it responds to a raging gram-negative bacterial infection.
It is a brilliant physiological paradox.
It really is. And to grasp it, we basically have to completely unlearn the way we were taught basic immunology in our first year of medical school.
Oh, yeah. The whole self versus non-self thing.
Right. We were all drilled on that classical model. The dogma was that the immune system is essentially a border patrol agency.
It ignores its own tissue self, and it violently attacks foreign invaders like bacteria or viruses non-self.
Precisely. But that classical model completely collapses the moment you step into the trauma bay.
Yeah.
A crushed pelvis is entirely self.
A devascularized segment of bowel is entirely self.
Right. Yet, as you described in your opening scenario, these injuries trigger an inflammatory cascade that is clinically indistinguishable from septic shock.
Because if the immune system only cared about non-self, a closed crush injury shouldn't trigger a fever and leukocytosis and profound vasodilation.
Exactly. And that discrepancy is what led to Polly Matzinger's danger model.
Which completely revolutionized our understanding of systemic inflammation.
It did. The danger model posits that the immune system, primarily the innate immune system, actually doesn't care if a molecule is foreign or native. It only cares if the environment is dangerous.
It's just looking for trouble.
Yeah, the innate immune system is constantly surveilling the extracellular fluid. Not checking passports to see if a protein is alien, but looking for the molecular debris of destruction.
And the way it differentiates between a normal, healthy tissue environment and a massive traumatic insult comes down entirely to the microscopic physics of cell death.
Yes. Walk me through that, because cells are dying in our bodies all the time, right?
Right. I have millions of cells dying right now as we speak, and I'm not going into ARDS.
Thankfully. because every day, millions of your cells undergo apoptosis.
The programmed cell death.
Exactly. It is a highly conserved, genetically programmed cell death. It requires energy. It is neat, orderly, and silent.
The cell essentially shrinks, right?
Yeah, it shrinks. Its DNA is systematically cleaved, and the plasma membrane undergoes a conformational change. The phosphatidylserine actually flips from the inner leaflet to the outer leaflet.
And that flipped phosphatidylserine acts as an eat me signal for local macrophages.
You got it. The macrophage engulfs the apoptotic body intact. The cell membrane never ruptures. None of the intracellular contents ever touch the extracellular fluid.
So the immune system remains completely quiet.
Silent. But blunt force trauma, penetrating trauma, ischemia, these forces don't ask the cell to politely undergo programmed apoptosis.
No, they definitely don't. Kinetic energy or crushing force causes necrosis, necroptosis, or pyroptosis.
Right. These are violent, chaotic, osmotically driven cell deaths. The cell swells, the organelles disintegrate, and crucially, the plasma membrane completely ruptures.
Reach the pop.
It pops. And when that membrane tears, the entire intracellular compartment, the cytoplasm, the nuclear material, all of it, is violently spilled into the extracellular space.
And that spilled intracellular material is what the immune system recognizes as danger.
Yes. To the immune system, the presence of intracellular proteins floating freely in the extracellular space is the ultimate alarm bell. It means a cell has been obliterated.
So how does it actually detect it?
The innate immune system detects this debris using evolutionarily primitive sensors called pattern recognition receptors, or PRRs.
Okay, PRRs.
Now, when a pathogen enters the body, these PRRs bind to PAMP's pathogen-associated molecular patterns.
Like lipopolysaccharide and gram-negative cell walls.
Right, exogenous stuff. But when a human cell violently bursts open from trauma, it releases DAMP's danger-associated molecular pattern.
Right, so known as alarmins, right?
Exactly, alarmins.
I want to pause here because this is where the evolutionary biology is staggering to me. A DAMP is an endogenous molecule. It is a normal piece of our own cellular machinery.
Yeah, it belongs there.
But it's in the wrong place at the wrong time. And despite one being bacterial and one being human, PAMs and DAMPs share incredibly similar conserved hydrophobic portions.
They do. They actually engage the exact same physical receptors on our macrophages.
It is a shared biological hardware. The macrophage doesn't have a separate alarm system for trauma and a separate one for infection.
Nope, just one master alarm system that detects tissue destruction, regardless of the cause.
Let's look at a couple of the most highly studied DAMPs that drive the clinical deterioration we see in our patients.
The big one is HMGB1, high mobility group MOX1.
Yeah, I see HMGB1 referenced constantly in the trauma literature as the primary driver of early systemic inflammation. But what actually is it? If it's a normal part of the cell, what is it doing before the trauma occurs?
Good question. So under normal, healthy conditions, HMGB1 is a highly conserved, non-histone, architectural chromatin-binding protein.
Okay, so it lives in the nucleus.
Exclusively inside the nucleus.
Yeah.
Its job is to bind to the minor groove of DNA, basically bending the double helix to facilitate gene transcription and DNA repair.
So it's tightly bound to the chromatin. It never sees the light of day.
Right. But when a cell undergoes necrotic death, say, a massive swath of skeletal muscle is crushed in an MVC with patient. The plasma membrane ruptures. The nuclear envelope disintegrates.
And HMGB1 is released into the extracellular space.
Suddenly, this DNA-bending nuclear protein is floating freely in the systemic circulation.
So if HMGB1 is usually locked inside the vault of the nucleus, its presence in the bloodstream is definitive proof to the immune system that the vault has been blown wide open.
That is exactly it. Extracellular HMGB1 is an exceptionally potent DAMP. When it hits the bloodstream, it actively drives the early systemic inflammatory response.
It pushes the immune system toward a Th1 pro-inflammatory phenotype, right?
Yeah. Furthermore, it acts as a massive chemoattractant. It establishes a chemical gradient that draws immature dendritic cells, monocytes, and neutrophils directly to the zone of injury.
Interestingly, if a cell dies neatly by apoptosis, the HMGB1 remains irreversibly cross-linked to the chromatin inside those neat little apoptotic bodies.
The alarm never sounds. It is specifically the violence of the necrotic cell death that sets HMGB1 free.
Wow. Another critical category of DAMPs we have to discuss are the heat shock proteins, or HSPs. Because these aren't just released by crushed cells, they are actively produced in response to the physiology of the trauma bay.
That's a really crucial distinction. Heat shock proteins are basically intracellular molecular chaperones. Their primary day job is to assist in the folding of newly synthesized proteins.
And refolding damaged ones.
Right. They're always present at baseline levels. But when a cell experiences severe physiological stress and in trauma, we are specifically talking about profound hypoxia, acidosis, and ischemia reperfusion injury, the cell desperately tries to survive.
So it drastically upregulates the transcription and translation of HSPs?
Yeah, it packs itself full of HSPs to protect its intracellular proteins from denaturing in that hostile acidic environment.
It's trying to armor up.
Exactly. And if that cell eventually lyses due to prolonged ischemia, it dumps a massive, artificially high load of these HSPs into the circulation.
And even more fascinating, I read that severely stressed cells that haven't died yet can actively secrete HSPs into the extracellular space via exosomes as a distress signal.
Yes. And once outside the cell, extracellular HSPs are highly immunogenic.
Yeah.
They bind directly to antigen-presenting cells, acting as massive danger signals that powerfully activate the innate immune cascade.
I always try to conceptualize this using an analogy of a highly integrated home security system.
Let's hear it.
So PAMPs, the bacterial molecules, are the sensors detecting a burglar actively smashing a window from the outside. That's the exogenous threat.
Right.
DAMPs, like HMGB1 and extracellular HSPs, are the internal sensors detecting a massive water pipe bursting inside the walls and flooding the living room. That's the endogenous threat.
I like that.
The central control panel doesn't care if the threat is external or internal. Either way, the exact same siren goes off, the exact same lights flash, and the exact same heavily armed first responders show up to the house.
That analogy perfectly sets up the actual hardware of that central control panel. The physical sensors on the surface of our immune cells that detect both the broken window and the burst pipe are the toll-like receptors, or TLRs.
TLRs are everywhere in the literature. But let's get into the weeds of how they actually function, because a DAMP floating in the blood doesn't cause ARDS by itself.
No, it has to physically dock.
Right. It has to physically dock with a TLR, and that docking has to transmit a signal into the nucleus of an immune cell.
This is where we bridge the gap between chemistry and clinical pathology. Toll-like receptors are transmembrane pattern recognition receptors expressed heavily on macrophages, dendritic cells, neutrophils, and even endothelial cells.
Currently we know of, 12 functional human TLR?
Yeah, 12. They have an extracellular leucine-rich repeat domain shaped like a horseshoe that physically binds the DAMP, and an intracellular TOL1 receptor domain that transmits the signal into the cytoplasm.
So an HNGB1 molecule from a crushed muscle cell floats through the capillary bed, bumps into a tissue macrophage, and physically binds into the horseshoe of a TLR4 receptor. What happens in the very next millisecond?
How does that physical connection turn into a cytokine storm?
Exactly.
It triggers one of two highly orchestrated intracellular signaling cascades. And understanding these is paramount because they dictate the entire phenotypic expression of systemic inflammation.
The primary cascade is the MyD88-dependent pathway, right?
Yes. This pathway is utilized by almost all the TLRs, with the notable exception of TLR3. When a DAMP binds to the extracellular domain, the intracellular domain undergoes a conformational change.
Which allows it to recruit an adapter protein called MyD88 from the cytoplasm.
Correct. MyD88 docks to the receptor inside the cell.
Then what?
MyD88 acts as a scaffold. It recruits and activates a series of kinases, specifically the interleukin-1 receptor-associated kinases, or IRAKs.
IRAKs, got it.
These IRAKs autophosphorylate and then recruit another factor called TRAF6. This creates a massive protein complex that eventually activates the IK complex.
I know we are deep in the alphabet soup here, but this specific step is the linchpin of everything we do in the ICU.
It absolutely is, because the IKK complex's sole job is to phosphorylate a small inhibitory protein called IKPOB.
And normally, IKPOB sits in the cytoplasm, holding on to a transcription factor called NF-kapOB, keeping it inactive.
Right. IKP is the safety on the gun. When IKK phosphorylates IKPOB, it marks it for degradation by the proteasome.
The safety is off.
The safety is off. NF-kappa-B is suddenly released. It immediately translocates through the nuclear pore, enters the nucleus of the macrophage, binds directly to the promoter regions of the DNA, and throws the master switch for inflammation.
It triggers the massive rapid transcription of our classic pro-inflammatory cytokines.
Interleukin-1, interleukin-6, interleukin-8, and tumor necrosis factor alpha.
Concurrently, doesn't the MyD88 pathway also activate the MAPK cascade?
Yes, the mitogen-activated protein kinase cascade, which stabilizes these newly formed cytokine RNAs so they can be translated into proteins efficiently.
So that is the MyD88 dependent pathway, but you mentioned an exception with TLR3.
Yeah, the MyD88 independent pathway, which is utilized by TLR3 and occasionally TLR4, instead of using MyD88 to trigger NF-kappa, this pathway uses a different adapter protein called TRIF.
And TRIF activates a different transcription factor, IRF3.
Exactly. IRF3 translocates to the nucleus and primarily induces the transcription of type 1 interferons.
Which are critical for antiviral responses.
Right, but the blunt force trauma response is overwhelmingly driven by the MyD88-dependent NF-kappa B-mediated cytokine explosion.
Okay, so to synthesize this first phase, kinetic energy shatters the cell membrane, bypassing neat apoptosis. Intracellular components like HMGB1 and heavily upregulated heat shock proteins are dumped into the extracellular fluid.
These act as dams.
They drift through the tissue, binding to toll-like receptors on local resident macrophages. This physical binding triggers the MyD88 adapter protein, kicking off a kinase cascade that ultimately degrades ICAPOB, releasing NF-kappa.
NF-kappa dives into the nucleus, unzips the DNA, and forces the macrophage to start printing thousands of inflammatory cytokines.
Which brings us directly into the systemic consequence of that microscopic event, the cytokine storm and the subsequent mobilization of our cellular cavalry.
The floodgates open.
Once those macrophages hit the print button, the local tissue is flooded with chemical messengers. And while this local reaction is biologically intended to clear the dead tissue and fight wound infections, the systemic spillover of these cytokines is exactly what destroys the patient's healthy, uninjured organs.
It is the ultimate double-edged sword of trauma biology. We cannot survive without this localized inflammatory response, but when the magnitude of the tissue injury is massive, the sheer volume of cytokines produced overwhelms the local tissue containment.
They spill into the systemic circulation.
Let's map the specific timeline of this cytokine storm because it is highly orchestrated, and the specific cytokines dictate the clinical vital signs you see on the monitor.
Okay, what hits first?
The hyperacute immediate responders are tumor necrosis factor alpha, or TNF-alpha, and interleukin-1 beta. These are synthesized and released within the first 1 to 2 hours following the traumatic insult.
They are the initial spark that sets the forest on fire.
And their systemic effects are profound. TNF-alpha and IL-1 beta travel to the hypothalamus and reset the thermoregulatory set point.
Causing the acute fever we see in trauma patients.
Right. They act on the liver to induce the acute phase response, which we will detail later. They cause profound systemic vasodilation, dropping the systemic vascular resistance and mimicking distributive shock.
And crucially, they act in an autocrine and paracrine fashion, right? They bind right back onto the macrophages and endothelial cells, commanding them to drastically upregulate the production of secondary cytokines.
Exactly. Which brings us to the subacute phase, dominated by the secondary mediators, IL-8, IL-12, IL-18, and the most critical one for our practice, IL-6.
I really want to drill down on IL-6 because the immunological literature provides a massive, actionable clinical pearl here for the practicing surgeon.
It really does.
The data consistently demonstrates that early circulating levels of IL-6 correlate directly in a linear fashion with the severity of the initial tissue destruction, usually quantified by the patient's injury severity score, or ISS.
Yes, IL-6 is the ultimate harbinger of clinical deterioration in the trauma population. Elevated IL-6 is not just a marker. It is biologically predictive.
Consistently high or rising levels of IL-6 in the first 24 to 48 hours are directly predictive of the development of ARDs, the progression to multiple organ failure, and the subsequent susceptibility to late sepsis.
When you are standing at that bedside watching a young patient with a severe pulmonary contusion rapidly descend into refractory hypoxemia on post-op day 3, you are watching the macroscopic clinical manifestation of an IL-6 storm destroying the alveolar capillary membrane.
But I want to push back here, or at least present a clinical dilemma that has always bothered me.
Okay, let's hear it.
We constantly talk about IL-6 as the primary bad actor, the match that starts the fire. It drives MOF. But when you dig deep into literature on autoregulation, it explicitly states that IL-6 also powerfully stimulates the release of anti-inflammatory mediators.
Oh, yes.
It triggers the production of IL-1 receptor antagonists, and it commands macrophages to synthesize and release prostaglandin E2, or PGE2, which is one of the most potent endogenous immunosuppressants in the human body.
It does.
So how can IL-6 be both the match that starts the fire and the chemical fire extinguisher? How do we reconcile that clinically?
It's a brilliant question, and reconciling it requires understanding the concept of immunological autoregulation, which ultimately leads to a state we call immunoparalysis.
Immunoparalysis.
The immune system is terrifyingly powerful. It evolved with the intrinsic knowledge that a runaway, unchecked inflammatory response is lethal to the host. Therefore, built into the very design of the pro-inflammatory cytokines are the triggers for their own suppression.
It is a massive biological negative feedback loop.
Exactly. When IL-6 spikes to massive levels following severe trauma, the central immune regulation realizes the system is overheating. It is causing too much collateral damage.
So IL-6 actively forces the release of PGE2 and interleukin-10.
Right. PGE2 rapidly suppresses T-cell proliferation, deactivates pro-inflammatory macrophages, and downregulates the production of TNF-alpha.
And IL-10 powerfully deactivates monocytes, preventing them from presenting antigens to T cells.
The body is desperately slamming on the emergency brakes.
So it's not a paradox at all. It's the body attempting to save itself from autodestruction.
Precisely. But here is the catastrophic clinical catch for the trauma surgeon. In severe trauma, the initial pro-inflammatory drive is so massive that the compensatory braking mechanism has to be equally massive to counter it.
The system slams on the brakes so hard that the car spins out.
That is a great way to put it. That massive release of PGE2 and IL-10, that profound immunoparalysis, is what leaves your patient incredibly vulnerable.
You successfully resuscitate them through the initial systemic inflammatory storm, but by post-op day 7, their macrophages are chemically deactivated.
Their cell-mediated immunity is paralyzed. They have no ability to mount a localized response to a new threat.
That is exactly why your trauma patient gets a devastating, overwhelming ventilator-associated pneumonia on day 7. Their immune system hasn't failed. It has been chemically subdued by its own aggressive autoregulatory mechanisms.
That perfectly explains the biphasic mortality curve we see in the SICU. Now, another major consequence of this cytokine milieu is a profound shift in the overall immune phenotype, specifically regarding our T helper cells.
Yes, the Th1 to Th2 shift.
Following major trauma, the production of interleukin-12, or IL-12, by monocytes and macrophages is significantly depressed.
And IL-12 is the critical cytokine responsible for driving naive T cells to differentiate into Th1 cells.
Exactly. Th1 cells are your classical pro-inflammatory cell-mediated warriors. They produce interferon gamma and activate cytotoxic macrophages to kill intracellular pathogens.
Because the trauma environment depresses IL-12, the immune system shifts away from that Th1 phenotype,
And instead, it favors the proliferation of Th2 cells.
which drive the humoral, anti-inflammatory, and allergic responses.
They produce IL-4, IL-10, IL-13, and transforming growth factor beta, TGF-beta. So we see this massive systemic shift from a Th1 cell-mediated state to a Th2 humoral and immunosuppressive state.
And the literature makes it abundantly clear. The magnitude of this Th1 to Th2 shift correlates strongly with adverse clinical outcomes and late mortality from opportunistic infections.
All right, so we have the systemic cytokine profile established. Now let's zoom in on what these cytokines are actually telling the effector cells to do.
Specifically, let's talk about the leukocyte recruitment cascade.
Because a neutrophil floating in the middle of the aorta doesn't do us any good.
Right. It has to leave the fast-flowing bloodstream, cross the endothelial barrier, and enter the damaged tissue.
This mechanism is happening in every capillary bed near the zone of injury, and unfortunately, in the capillary beds of innocent bystander organs like the lungs and the kidneys.
The mechanism of extravasation is an elegant, highly conserved four-step sequence. It requires precise spatial and temporal orchestration between the circulating neutrophil and the vascular endothelium.
Let's walk through it. Step 1 is capture and tethering. Imagine a neutrophil flying through a capillary at incredibly high velocity.
The local vascular endothelium has been activated by the systemic release of TNF-alpha and IL-1 beta from the trauma site.
Right. The neutrophil constitutively expresses a molecule on its surface called L-selectin. As it flies by, this L-selectin reaches out and lightly catches specific glycoprotein ligands on the activated endothelial cells.
It's a very low affinity bond, right? It breaks almost instantly due to the sheer force of the flowing blood, but it catches just long enough to slow the neutrophil's momentum.
It's like a grappling hook, lightly catching the edge of a wall, just slowing the cell down.
Right.
Which sets up step 2, rolling.
Rolling.
The tethering slows the leukocyte down enough for the more robust endothelial selectins to take over.
Right.
The endothelial cells, stimulated by the injury and histamine release, rapidly exocytose preformed P-selectin from specialized storage granules called Weibel-Palade bodies.
Weibel-Palade bodies, and simultaneously, driven by NF-kappa B transcription, the endothelium synthesizes and expresses E-selectin de novo.
Exactly. These E and P selectins bind to a heavily glycosylated carbohydrate structure on the rolling leukocyte called PSGL1, P-selectin glycoprotein ligand 1.
These bonds are fascinating because they're catch bonds. They actually require a certain amount of shear stress from the flowing blood to maintain their grip.
Yes, the bonds form rapidly at the leading edge of the cell and break rapidly at the trailing edge, causing the leukocyte to physically tumble and roll along the endothelial wall against the current of the blood.
It's tumbling along, sensing the environment. And that rolling brings us to step 3, firm adhesion.
The rolling leukocyte is constantly sampling the local environment.
When it rolls through a concentrated gradient of local chemokines, specifically IL-8 presented on the endothelial surface, it triggers an inside-out intracellular signaling cascade within the neutrophil.
That inside-out cascade is key. It causes a massive rapid conformational change in the neutrophil's membrane integrins, specifically the beta-1 and beta-2 integrins like MAC1 and LFA1.
The integrins shift from a bent low-affinity state to an extended high-affinity state.
Right. These extended integrins bind tightly and irreversibly to receptors on the endothelial wall, primarily ICAM1, intercellular adhesion molecule 1.
This creates a completely stable, shear-resistant adhesion. The cell stops rolling immediately. It flattens out. It is locked in place.
And that lock sets up the final step, transmigration or diapedesis.
The arrested leukocyte literally squeezes its nucleus and cytoplasm between the tight junctions of the endothelial cells, crossing the physical barrier to enter the extravascular tissue space.
This diapedesis is mediated by PCAM1, platelet endothelial cell adhesion molecule, which acts almost like a molecular zipper, actively pulling the leukocyte through the intercellular junctions.
So now, the neutrophils have successfully arrived in the damaged tissue. Their biological mandate is to clear necrotic debris and destroy any contaminating bacteria.
But here's where we must discuss the devastating collateral damage. Because neutrophils do not use scalpels to clear tissue, they use biological explosives.
When a neutrophil activates in the tissue bed, it undergoes profound degranulation. It dumps its intracellular vesicles into the extracellular matrix.
These granules contain highly potent extracellular proteases, specifically neutrophil elastase, matrix metalloproteinases, or MMPs, and cathepsin G.
These enzymes are designed to digest connective tissue. They indiscriminately cleave structural proteins, collagen, elastin, and the extracellular matrix scaffolding. They literally digest the local tissue architecture.
And unfortunately, it gets much worse than just proteases. Alongside the enzymatic destruction, the activated neutrophils unleash the respiratory burst.
This is the massive, rapid generation of reactive oxygen species, or ROS.
The biochemistry of the respiratory burst is a weapon of mass destruction. It is driven by an enzyme complex called NADPH oxidase.
In an activated neutrophil, the subunits of NADPH oxidase assemble on the phagosome membrane or the plasma membrane.
This complex steals electrons from intracellular NADPH and transfers them directly to molecular oxygen, generating massive quantities of the superoxide anion.
But superoxide is just the precursor, right?
Yeah. Right. An enzyme called superoxide dismutase rapidly converts that superoxide into hydrogen peroxide.
Hydrogen peroxide is toxic, but it is relatively stable.
However, in the chaotic, iron-rich environment of a tissue hematoma, the Fenton reaction kicks in. Free iron catalyzes the reduction of hydrogen peroxide into the highly toxic, wildly reactive hydroxyl radical.
And simultaneously, we have reactive nitrogen species joining the fight.
Yes. Cytokines powerfully upregulate inducible nitric oxide synthase, or INS, in macrophages and endothelial cells, forcing them to produce massive amounts of nitric oxide.
When nitric oxide reacts with the superoxide anion, it forms peroxynitrate, an incredibly damaging reactive nitrogen species.
When you combine these hydroxyl radicals, the peroxynitrite and the proteases, you get severe lipid peroxidation.
The reactive species literally rip electrons off the lipid bilayers of healthy cells, causing cell membrane disintegration, profound DNA damage, and widespread destruction of the local endothelial cells.
And what does all this microscopic biochemical warfare mean for you, the surgeon, standing at the bedside trying to manage a fluid balance?
It means generalized capillary leak syndrome.
When the neutrophils marginate in the pulmonary capillaries or the splanchnic beds, and they unleash these proteases and reactive oxygen species. The tight junctions of the endothelial cells are digested.
The endothelial barrier fundamentally breaks down. The massive volume of crystalloid and blood products you are aggressively pumping into the patient's intravascular space simply pours right out of the porous vessels and into the interstitium.
This is exactly why your patient's bowel is massively edematous, weeping fluid, and preventing primary abdominal closure.
This is why their lungs are heavy and stiff, filled with proteinaceous fluid. This is why they are 20 liters positive and look like the Michelin Man on post-op day 4.
It is the direct protease and ROS-mediated physical destruction of the endothelial barrier.
Which serves as the perfect biological bridge to our next major clinical concept. Because that severe inflammation causing the capillary leak doesn't just affect fluid shifts.
It fundamentally and structurally alters the blood vessel walls themselves,
Leading directly to the complex consumptive coagulopathies that you battle during massive transfusions. Let's move into the tangled web of immunothrombosis and the destruction of the glycocalyx.
This concept of immunothrombosis completely shifted my understanding of trauma-induced coagulopathy.
Teleologically, it makes perfect evolutionary sense to physically combine hemostasis with pathogen elimination. If a predator bites you or you suffer a massive open wound, you have a physical hole in a blood vessel.
That hole is bleeding, but it is also the exact doorway a pathogen will use to enter the bloodstream.
So the body evolved a redundant, overlapping system where the innate immune response and the coagulation cascade are deeply physically, chemically, and spatially intertwined.
The clot isn't just a cork to stop bleeding, it is a physical barrier to trap bugs.
They are essentially two sides of the exact same biological coin. We used to think of coagulation and immunity as distinct chapters in a textbook, but they occur simultaneously.
Let's look at how the effectors of the innate immune system directly force coagulation. Monocytes, in response to binding those DAMPs and PAMPs we discussed earlier, actively synthesize and express tissue factor on their cell surfaces right at the site of injury.
This circulating tissue factor forcefully and directly activates the extrinsic coagulation pathway, independent of the subendothelial tissue factor exposed by the injury itself.
But perhaps the most visually striking and clinically dangerous example of this overlap is the formation of NETs neutrophil extracellular traps.
The first time I read about nets, it sounded like a science fiction mechanism. Walk us through exactly what a net is and how a cell performs this function.
It is a phenomenal, highly specialized form of cell death called netosis. When a neutrophil is highly activated by severe trauma or overwhelming infection, it realizes that phagocytosis-eating bacteria one by one is insufficient.
So it deploys a final suicidal weapon.
The neutrophil's nucleus undergoes profound changes. Peptidyl-larginine deaminase, or PAD4 enzymes, citrullinate the core histones, which abolishes their positive charge.
This causes the tightly coiled chromatin to massively decondense and unravel.
The nuclear envelope disintegrates, allowing this unraveled DNA to mix with the highly toxic antimicrobial proteins in the cytoplasm, specifically neutrophil elastase and myeloperoxidase.
Finally, the plasma membrane ruptures, and the neutrophil literally extrudes its own unraveled DNA, coated in these toxic granular proteins, into the extracellular space and the flowing bloodstream.
It forms a massive physical, sticky web.
The primary evolutionary goal is to throw a net to physically ensnare, immobilize, and kill circulating pathogens. But as we see in severe trauma, those nets do significantly more than just catch bacteria.
Exactly. And this is where the coagulopathy begins. Those nets are composed of extracellular DNA. DNA is highly negatively charged.
That massive negatively charged surface acts as a potent contact activation surface for the coagulation cascade.
It directly binds and autoactivates factor 12, forcefully kicking off the intrinsic coagulation cascade right in the middle of the flowing blood.
Furthermore, the structural lattice of the nets physically binds von Willebrand factor, it binds circulating tissue factor, and it actively recruits and aggregates passing platelets.
So millions of neutrophils are throwing massive DNA nets into the circulation that not only catch debris, but serve as a physical scaffold to rapidly build massive blood clots.
It's a brilliant defense mechanism for containing a small, localized abscess. But in the setting of major trauma, when you have a massive damp release, systemic inflammation, and intravascular net formation occurring simultaneously in capillary beds all over the body,
This immunothrombosis becomes lethal.
This mechanism actively drives the disseminated intravascular coagulation, or DIC phenotype we see in severe trauma.
You get millions of intravascular microthrombi plugging up the capillary beds of the kidneys, the lungs, and the liver, causing profound ischemic cellular damage.
And simultaneously, building all of those microscopic clots consumes massive quantities of your circulating platelets, fibrinogen, and clotting factors.
Which leaves the patient profoundly coagulopathic and bleeding from every IV site and surgical incision.
It is the definition of consumptive coagulopathy, and the foundation upon which all this catastrophic, inappropriate coagulation occurs is the injured endothelium itself.
We have to discuss a vital vascular structure that has moved to the forefront of modern trauma resuscitation, the endothelial glycocalyx.
The glycocalyx is paramount. We used to think of endothelial cells just like smooth tiles lining a pipe, but they aren't smooth.
Far from it. The glycocalyx is a microscopic, highly complex, gel-like endovascular layer that coats the luminal surface of every healthy blood vessel in the body.
It is composed of membrane-bound proteoglycans and glycoproteins, creating a thick, slick, protective web.
The primary structural backbone of this protective layer is a heparin sulfate proteoglycan called syndican-1.
Under normal physiological conditions, the glycocalyx is what maintains vascular homeostasis. It physically covers and hides the endothelial adhesion molecules, like ICAM, and the selectin-so-passing leukocytes can't see them.
It houses antithrombin III and tissue factor pathway inhibitor, making the vessel wall inherently anticoagulant.
And it provides the oncotic gradient that prevents plasma water from leaking into the interstitium. It is the ultimate non-stick protective Teflon coating.
But the trauma literature clearly demonstrates that under the specific conditions of profound hemorrhagic shock, hypoperfusion, and systemic sympathoadrenal activation, that structural backbone syndican-1 is actively attacked.
Yes. The shock state causes endothelial cells to release shedoses, specifically metalloproteinases, which rapidly cleave syndican-1 right at the cell membrane.
The entire glycocalyx is physically shed, degraded, and washed away into the bloodstream. The vessel loses its Teflon coating.
And the consequences of losing that coating are immediate and catastrophic.
When the syndican-1 sheds, the underlying endothelial cells are laid bare. All of those pro-inflammatory adhesion molecules, the P-selectins, the E-selectins, the ICAMs we discussed earlier, are suddenly naked and exposed to the flowing blood.
The endogenous anticoagulants are washed away. You immediately transform the entire vascular tree from a slick antithrombotic tube into a highly prothrombotic leukocyte-grabbing surface.
This generalized endotheliopathy actively drives the massive systemic consumption of coagulation factors and the massive margination of neutrophils.
This is exactly why I wanted to dive into this molecular biology, because this explains the why behind our most critical clinical protocols.
10 or 15 years ago, we resuscitated trauma patients with massive amounts of crystalloid and packed red blood cells.
But modern massive transfusion protocols, our MTPs, emphasize the early, aggressive, and preemptive use of plasma, pushing toward a 1.1.1 ratio of plasma to platelets to red cells.
When I am ordering coolers of plasma for a patient in hemorrhagic shock, I am not just giving FFP to replace floating clotting factors in a vacuum.
You are actively trying to chemically resuscitate the injured endothelium. The plasma provides the specific oncotic proteins, the fibrinogen, and the molecular substrates necessary to rebuild that shattered glycocalyx and turn off the generalized endotheliopathy.
We're treating the blood vessel wall itself, trying to restore that non-stick surface to halt the consumptive DIC process.
That is the exact physiological rationale. You are treating the pipe, not just the fluid inside it.
And to complete this web of immunothrombosis, we must briefly mention how the complement and kallikrein-kinin systems amplify this chaos.
When endothelial damage occurs and the negatively charged surfaces are exposed, either by shed glycocalyx or by NETs factor 12 is activated to factor 12.
Factor 12 doesn't just trigger the intrinsic coagulation pathway. It simultaneously activates prekallikrein to kallikrein.
Kallikrein cleaves high molecular weight kininogen to produce massive amounts of bradykinin.
Bradykinin is an extraordinarily potent vasodilator and powerfully increases vascular permeability, massively exacerbating the third spacing and distributive shock.
And the complement cascade is right there beside it. The tissue ischemia, the exposed basement membranes, the proteases released by neutrophils, all of these trigger the cleavage of complement proteins, specifically generating the anaphylatoxins C3A and C5A.
C5A is particularly nasty in this setting. It amplifies the systemic inflammation by recruiting thousands more phagocytes to the area.
It actively induces cellular apoptosis in healthy bystander cells, and it strongly activates platelets, feeding right back into that dangerous loop of immunothrombosis.
It is a devastatingly self-amplifying cycle. The DAMPs trigger the cytokines trigger the neutrophil rolling, the neutrophils unleash ROS and NETs.
And the nets and shock destroy the glycocalyx and activate coagulation, which causes more ischemia, which releases more DAMPs.
So having mapped this microscopic intracellular chaos, we now need to pull the camera back. We need to look at how these molecular pathways coalesce into the macroscopic clinical syndromes that you manage in the SICU.
Let's transition to the clinical phenotypes. We've described the cellular battlefield. Let's look at the systemic war.
I want to start with the immediate, massive systemic response to all these cytokines hitting the major organs, specifically the liver.
We see the metabolic consequences of this on our daily morning labs, known as the acute phase reaction. Within hours of the initial kinetic injury, driven primarily by the massive surge of IL-6, IL-1, and TNF-alpha, the biosynthetic profile of the hepatocyte is completely and forcefully reprogrammed.
Reprogrammed is the perfect word. Under normal, steady-state conditions, the liver is happily chugging along synthesizing baseline plasma proteins, primarily what we call negative acute phase proteins.
The most prominent are albumin, prealbumin, and transferrin. They maintain oncotic pressure and transport nutrients.
But when the Kupffer cells, the resident macrophages of the liver, sense that massive systemic cytokine storm rolling in from the trauma site, they sound the alarm.
The liver abruptly, actively halts the transcription and translation of those negative acute phase proteins.
It reallocates all of its available amino acids, ribosomes, and energetic capacity to synthesize a completely different set of molecules, the positive acute phase proteins.
I think this is a critical point that is often misunderstood. When you are looking at your trauma patients' morning labs on post-op day 2, and you see their serum albumin has tanked from a healthy 4.0 down to 1.8, your immediate thought is often, well, they're third spacing, the albumin is leaking out of the capillaries, or it's just dilutional from the 10 liters of crystalloid we gave them.
And while dilution and capillary leak play a role, the primary physiological reason for that drop is that the liver purposefully, actively stopped making it.
The liver turns off the albumin factory so it can prioritize the synthesis of proteins essential for surviving the inflammatory storm, primarily C-reactive protein, or CRP, and serum amyloid A, SAA.
Exactly. It is a purposeful reallocation of resources, and the sheer volume of production is staggering. CRP levels can surge up to 1,000-fold above baseline within 24 to 48 hours.
And CRP serves a vital targeted function. It actively binds to the exposed chromatin and phosphocholine of necrotic cells.
It acts as an opsonin, flagging the debris for macrophage clearance, and it likely helps mask those exposed nuclear antigens to prevent the development of secondary autoimmune reactions down the line.
SAA is equally fascinating. SAA physically binds to the third fraction of high-density lipoprotein, HDL3, completely displacing apolipoprotein AI to become the dominant apolipoprotein on the HDL molecule during acute inflammation.
This conformational change radically enhances the ability of HDL3 to bind to activated macrophages.
It essentially creates a massive fleet of garbage trucks that rapidly scavenge the massive amounts of toxic cholesterol and lipid debris released from shattered cell membranes.
That scavenged cholesterol is then safely transported to the liver and recycled for future tissue repair.
It is an incredible salvage and cleanup operation. Now, this entire overwhelming systemic reaction, the tachycardia, the tachypnea to blow off acid, the fever driven by PDE2 in the hypothalamus, the leukocytosis from bone marrow demargination, we categorize this clinically as Systemic Inflammatory Response Syndrome, or SIRS.
We all know the classical 1991 criteria. But what the foundational immunology highlights is that SIRS is driven entirely by the endogenous inflammatory process, completely independent of infection.
If an infection is proven to be the trigger, then it's sepsis. But our trauma patients are in profound sterile SIRS.
Yes, and paralleling that SIRS response, often initiated almost simultaneously, is CARS, the Compensatory Anti-Inflammatory Response Syndrome.
We touched on this when discussing the autoregulation of IL-6. CARS is the body's massive systemic attempt to rein in the destructive potential of SIRS.
It is driven by the massive systemic release of IL-10, TGF-beta, and PGE2, which actively suppress T-cell function and deactivate macrophages.
When SIRS and CARS are perfectly balanced, the patient exhibits a controlled fever, clears the dead tissue locally, the inflammation subsides, and they heal smoothly.
But when they are unbalanced, clinical disaster strikes. If SIRS predominates unchecked, The patient dies early of cardiovascular collapse, massive capillary leak, and multiple organ failure.
If CARS overwhelms the system, the patient survives the initial trauma but enters a state of profound, prolonged post-traumatic immunosuppression.
They survive the first week, only to die in week 3 from opportunistic nosocomial infections, because their immune system is chemically paralyzed.
The literature also details Mars mixed antagonist response syndrome. This describes the patient who exhibits intermittent back-and-forth surges of both SIRS and CARS phenotypes.
They run a fever and require pressors on Monday. They stabilize by Wednesday. They drop their white count and get a VAP on Friday. And then they surge back into a SIRS response.
It reinforces the reality that these aren't just binary on-off switches. They are dynamic continuums, like a complex rheostat, constantly reacting and adjusting the systemic levels of pro- and anti-inflammatory mediators based on real-time feedback.
But perhaps the most clinically actionable, game-changing concept in the entire text regarding these phenotypes is the two-hit model of post-injury multiple organ failure.
The two-hit model is an absolute conceptual necessity for every practicing trauma surgeon to internalize, because it completely dictates your operative decision-making.
The cellular concept is that the initial trauma, the high-speed car crash, the blast injury, the crush, is the first hit.
This first hit triggers the massive systemic release of DAMPs and the subsequent cytokine storm we just spent 30 minutes detailing.
This massive systemic signaling profoundly primes the innate immune system. The circulating neutrophils are activated. They rapidly marginate and adhere to the endothelial walls of the pulmonary, splanchnic, and hepatic capillary beds.
Inside those neutrophils, the NADPH oxidase complex is assembled on the membrane, loaded, and waiting. The intracellular elastase granules are mobilized to the cell periphery.
The entire innate immune system is cocked and loaded, physically parked in the vulnerable organ beds, waiting for a final trigger.
And if the patient avoids a second hit, if they remain stable, that primed state slowly, naturally resolves. The neutrophils undergo apoptosis and are cleared.
But if a second hit occurs during that window of extreme vulnerability, that primed immune system unleashes a completely disproportionate, exaggerated, and often lethal inflammatory reaction.
The marginated neutrophils simultaneously degranulate and unleash their respiratory burst directly into the delicate alveolar capillary membranes of the lungs, and the patient goes into fatal AR deaths.
Now, the literature lists endogenous second hits like an episode of severe hypoxia, a period of profound cardiovascular instability or recurrent shock, or the massive ischemia-reperfusion injury of a clamped vessel.
And it lists exogenous second hits like massive blood transfusions, missed injuries, or severe secondary infections.
But I have to interject here because looking at this from a surgical timing perspective, we have to acknowledge a harsh reality. As surgeons, we are the second hit.
We absolutely are.
Okay.
When we take a stabilized, heavily primed trauma patient back to the operating room on hospital day 2 for a scheduled massive 5-hour definitive femur fixation, or to wash out a massive open abdominal wound, the physical surgical incision, the intramedullary reaming, the profound tissue handling, all of that releases a massive fresh wave of new DAMPs into the circulation.
We are actively physically pulling the trigger on that primed immune system.
That is the exact harsh clinical reality. Your surgical intervention, no matter how technically flawless, is a massive exogenous second hit.
In understanding this precise cellular mechanism, understanding the kinetics of neutrophil margination and priming is what completely changed the paradigm of surgical timing in our field.
It is the entire biological and physiological basis for damage control orthopedics and damage control surgery.
You do not subject a massively primed, hemodynamically borderline vulnerable patient to a 4-hour definitive intramedullary nailing of a femur on post-op day 2. You recognize that the patient is in the peak of their SIRS response.
So you place a temporary external fixator, a minimal rapid surgical procedure that acts as a very small, tolerable second hit.
You stabilize the bone, you stop the marrow bleeding, and then you get out of the OR. You allow the systemic SIRS response to cool off over several days. You wait for the marginated neutrophils to clear.
And you only return to the OR for the definitive major intramedullary nailing when the immune system has deprised, the IL-6 levels have fallen, and the patient has entered a stable anabolic recovery phase.
It is cellular biology dictating operating room logistics. It's brilliant.
But let's move down the timeline. Let's say your patient survives the massive first hit. Your damage control timing was perfect, and they survived the surgical second hits without tipping into fulminant MOF.
But they just stay in the ICU. It's day 21. They are tracheostomized. They have a PEG tube. They have recurrent low-grade bacteremias from lines and indwelling catheters.
Their wounds simply will not heal. Their granulation tissue is pale and friable, and they are profoundly wasting away, losing massive amounts of lean muscle mass despite aggressive, calculated enteral feeding.
The critical care literature has defined a specific, distinct phenotype for this exact patient: PICS, or persistent inflammation, immunosuppression, and catabolism syndrome.
PICS is undeniably the modern plague of the surgical ICU. As our early resuscitation strategies, our massive transfusion protocols, and our damage control techniques have drastically improved, significantly fewer patients die in the first 48 hours from exsanguinating hemorrhage or refractory shock.
Instead, they survive that initial insult only to develop PICS.
The defining clinical characteristics are right there in the name: persistent, smoldering, low-grade inflammation, profound, unbreakable immunosuppression leading to relentless sepsis recidivism, and massive refractory protein catabolism.
The criteria to identify these patients clinically are highly specific. An ICU stay greater than 14 days. Persistent systemic inflammation, often marked by a CRP greater than 50 or an absolute neutrophil count that won't normalize.
Persistent severe immunosuppression, defined by a total lymphocyte count of less than 0.8.
And a profound catabolic state, marked by a serum albumin consistently less than 3.0, a prealbumin less than 10, or massive unintentional weight loss exceeding 10% of their body mass during their hospitalization.
So if the patient is fully resuscitated and we've achieved definitive source control of any active infections, what is driving this chronic, miserable systemic state? The recent immunological research points the finger at a very specific cellular culprit: myeloid-derived suppressor cells, or MDSCs.
Yes. The discovery and characterization of MDSCs completely illuminated the pathophysiology of PICS. MDSCs are a highly heterogeneous population of immature myeloid cells. They are the early precursors to macrophages, granulocytes, and dendritic cells.
Under normal, healthy physiological conditions, they exist in very small numbers. They act locally right at the site of inflamed tissue to suppress the acute inflammatory response once an infection is cleared.
They are the local peacekeepers ensuring the immune system turns off.
But in the setting of severe catastrophic trauma and prolonged SIRS, the bone marrow is massively, continuously stimulated. This population of immature MDSCs pathologically expands.
They rapidly leave the bone marrow, they leave the localized zone of injury, and they systematically disseminate throughout the entire body. They take up residence in the spleen, the lymph nodes, and the systemic circulation.
And once they go systemic, they enforce a brutal, generalized, unbreakable suppression of both the innate and adaptive immune systems. The literature details exactly how they execute this suppression, and it is a multi-pronged attack.
They have direct chemical mechanisms.
Mm.
They pump out massive amounts of immunosuppressive nitric oxide and reactive oxygen species that directly paralyze surrounding T cells.
They express arginase, which actively depletes the microenvironment of arginine, a vital amino acid we will discuss in a moment.
They secrete massive quantities of the immunosuppressive cytokines TGF-beta and IL-10. And incredibly, they express FAS ligand on their surface, allowing them to physically bind to active effector T cells and directly induce cellular apoptosis.
They literally force the fighting T cells to commit suicide.
And they utilize powerful indirect mechanisms as well. MDSCs actively secrete factors that force the massive expansion of regulatory T cells, Tregs, which further suppress the immune environment.
They actively skew resident tissue macrophages away from the pro-inflammatory M1 phenotype toward the suppressive tissue remodeling M2 phenotype, and they simultaneously inhibit the cytotoxicity of natural killer cells.
The patient's entire immune system is essentially placed in an inescapable pharmacological straitjacket by these massively expanded MDSCs. They cannot mount an effective response to a new pathogen, which is why they succumb to recurrent, low-virulence nosocomial infections week after week.
Which naturally leads us to the daily practical dilemma we face on rounds every single morning: nutrition and resuscitation. Because if PICS is defined partly by severe, unbreakable protein catabolism, the immediate clinical imperative is how we feed and physically support these chronically critically ill patients to alter their immune destiny.
I want to dive into the specific nutritional debates we have in the ICU, using the fundamental biology as our guide.
Let's start with the daily debate over specific amino acid formulations. We write orders for total parenteral nutrition or advanced enteral feeds every day, and there is a constant discussion about enriching these feeds with specific amino acids. To the naked eye, protein is just protein, right? We just need to give them enough calories and nitrogen to stop the muscle wasting. Why does the cellular biology demand specific, targeted amino acid supplementation in these massively traumatized patients?
Because in the setting of major trauma, protein is absolutely not just protein. The metabolic pathways of the patient are fundamentally altered. The literature explicitly highlights the critical roles of glutamine, alanine, and arginine.
Let's look at the physiology of glutamine and alanine first. During severe stress, sepsis, and prolonged cirrhosis, the body desperately needs massive amounts of energy and specific building blocks to fuel the hyperactive immune system and the massive liver protein synthesis we discussed.
To get these, the body mercilessly cannibalizes its own skeletal muscle. The muscle undergoes massive, rapid catabolism.
Amazingly, while glutamine and alanine only constitute about 6 to 8% of the structural protein in normal, healthy skeletal muscle, they account for over 70% of the total free amino acids released into the bloodstream during this trauma-induced muscle breakdown.
The body is specifically, preferentially mobilizing these two exact molecules.
It's tearing down the house just to harvest a very specific type of copper wire. Why? What is the profound systemic demand for glutamine?
Glutamine becomes a conditionally essential amino acid during severe stress because it serves two absolutely critical physiological roles. First, it is the primary preferred metabolic fuel source for rapidly dividing cells, specifically the enterocytes that line the mucosal barrier of the gastrointestinal tract and the proliferating leukocytes of the immune system.
If glutamine is depleted, and it rapidly is in massive trauma, the enterocytes literally starve to death. The mucosal barrier of the gut breaks down, the tight junctions fail, and you get massive unchecked bacterial translocation of endogenous gut flora directly from the intestinal lumen into the portal circulation and the mesenteric lymphatics.
This feeds right back into the SIRS cascade. Second, glutamine is an absolutely necessary rate-limiting molecular precursor for the synthesis of glutathione.
Glutathione is arguably the most critical endogenous intracellular antioxidant in the human body. Without glutamine, the cells cannot produce glutathione, and they are left completely defenseless against the massive reactive oxygen species storm unleashed by the neutrophils.
That perfectly explains the rationale behind glutamine supplementation protocols in the burn unit and the SICU. What about arginine? We constantly hear about arginine deficiency in these chronically ill trauma patients.
Arginine depletion ties directly back to our discussion of those pathological myeloid-derived suppressor cells. Remember that highly activated MDSCs and degranulating granulocytes heavily express the enzyme arginase.
Arginase rapidly and voraciously consumes and degrades all available circulating arginine in the plasma. The degree of arginine depletion is directly, linearly proportional to the severity of the initial traumatic insult and the magnitude of the MDSC expansion.
The catastrophic problem is that your T cells, the critical effectors of your adaptive immune system, absolutely require a rich supply of exogenous arginine for activation, for rapid clonal expansion, and for the proper development of memory phenotypes.
The T cell receptor physically relies on arginine to function. If the massive swarm of MDSCs actively degrades all the arginine in the microenvironment, the T cells are metabolically starved and completely paralyzed. It's one of the primary targetable mechanisms of PICS.
So the biological rationale for arginine supplementation is to flood the system, overwhelming the arginase enzymes, and finally give the T cells the metabolic fuel they need to wake up and fight off the nosocomial infections.
Exactly. Now, to be clinically accurate, while the cellular physiology is crystal clear, the literature notes that large-scale clinical trials on isolated arginine supplementation in general sepsis have been somewhat mixed, likely due to heterogeneous patient populations. So it remains a highly debated practice.
But the underlying biological rationale in the severely traumatized PICS phenotype patient is incredibly sound. Let's pivot from macronutrients to micronutrients. We've all seen the standard trauma vitamin pack ordered on admission, a massive doses of zinc, selenium, ascorbic acid. There's always a skeptic on rounds who says, are we just making very expensive vitamin-rich urine in the ICU? Or do these trace elements actually alter the immunological trajectory of a patient in profound shock?
They absolutely unequivocally matter. To understand why, you have to think right back to our deep dive into the neutrophil respiratory burst. Think about the NADPH oxidase complex assembling on the membrane. Think about the massive generation of the superoxide anion.
Think about the Haber-Weiss reaction and the iron-catalyzed Fenton reaction generating those lethal tissue-destroying hydroxyl radicals.
The only mechanism the human body possesses to mitigate that catastrophic generalized tissue and endothelial damage is its endogenous antioxidant enzyme systems, specifically enzymes like superoxide dismutase, glutathione peroxidase, and catalase.
But these massive complex enzymes do not function in a vacuum. They absolutely require specific trace element cofactors to catalyze the reduction of those toxic radicals.
Selenium is the required cofactor for glutathione peroxidase. Zinc, copper, and manganese are the required cofactors for the different isoforms of superoxide dismutase.
And vitamin C and E serve directly as essential biochemical electron sinks. They physically absorb the unpaired electrons from the free radicals, neutralizing the oxidative damage before it rips apart the lipid bilayer of the endothelial cells.
And because the magnitude of the ROS generation in major trauma is so astronomically high, the body rapidly burns through its entire baseline stockpile of these cofactors, trying to put out the fire.
Yes. The literature demonstrates clearly that severe trauma and major burn patients have profoundly depleted serum and intracellular levels of these essential cofactors, and the depletion is directly proportional to their injury severity score. The body consumes its entire biochemical shield.
The data highlights that aggressive, targeted, high-dose micronutrient supplementation has been explicitly demonstrated to significantly decrease wound healing time, decrease the incidence of nosocomial pneumonia, and decrease the overall magnitude of systemic inflammation in severe burn and trauma patients.
They are absolutely not just expensive vitamins. They are the required biochemical ammunition for the enzymes protecting the glycocalyx and the endothelium. Okay, this brings me to a final overarching physiological question. We have spent an hour discussing how the inflammation violently starts, how it cascades systemically, how it destroys tissues, and how it actively suppresses the immune system to the point of PICS.
But if a patient actually survives, if they clear the necrotic tissue, heal their surgical incisions, and successfully transfer out of the ICU to the floor, how does the human body ever actually turn this massive inflammatory machine off?
For decades, I think we subconsciously assumed that the resolution of inflammation was just a passive process. We assumed that the damps eventually washed out of the system, the cytokines slowly degraded, the neutrophils died of old age, and the fire just eventually burned itself out.
But the modern literature highlights a complete paradigm shift regarding resolution. It is a massive paradigm shift. The transition from a profound catabolic inflammatory state to an anabolic healing and resolving state is absolutely not passive.
It is a highly active, biochemically driven, energy-dependent process. The immune system doesn't just run out of gas, it receives a specific, powerful molecular command to stop fighting and start rebuilding.
Active resolution. What is the specific cellular mechanism that commands that shift?
It involves a profound, highly coordinated shift in localized lipid chemistry within the damaged tissue. During the acute hyperinflammatory phase of trauma, arachidonic acid released from damaged cell membranes is rapidly metabolized by cyclooxygenase, or KOX, and lipoxygenase LOX enzymes.
This produces massive quantities of our classic pro-inflammatory lipid mediators,
Yeah.
prostaglandins and leukotrienes. These are the molecules driving the massive neutrophil influx and the capillary leak.
But as the initial damps are cleared and specific temporal triggers are met within the tissue microenvironment, the cellular enzymatic machinery undergoes a radical class switch. The exact same enzymes, the lipoxygenases, actively shift their metabolic pathways.
Instead of utilizing arachidonic acid to produce pro-inflammatory leukotrienes, they begin utilizing essential omega-3 fatty acids like EPA and DHA to synthesize a completely different, highly specialized class of lipid mediators.
The literature specifically names these molecules: resolvins, protectins, and lipoxins.
Resolvins and protectins. What do these specific molecules actually do to turn off the SIRA's response?
They are the ultimate biological peacekeepers. They don't just competitively block inflammatory receptors, they actively command the cessation of the immune response. When resolvins and protectins are synthesized in the tissue, they halt any further neutrophil extravasation.
They actively seal up the endothelial junctions to stop the capillary leak. Most importantly, they bind directly to the resident tissue macrophages and completely reprogram them.
They command the macrophages to stop producing pro-inflammatory cytokines, and they trigger a massive upregulation of a process called efferocytosis.
Efferocytosis is the highly efficient, non-inflammatory phagocytosis and clearance of all the dead, spent, and apoptotic neutrophils cluttering the tissue bed. The macrophages rapidly eat the dead neutrophils, clear the debris field, and then simultaneously release massive amounts of tissue remodeling factors, VEGF, and anti-inflammatory cytokines to initiate the physical rebuilding of the extracellular matrix.
That is an incredibly elegant biological system. It is not a passive burnout, it is an active, commanded reconstruction. Which brings us perfectly to the synthesis and our final actionable takeaways for your daily practice in the trauma bay and the SICU. We have covered a massive amount of cellular ground today. We started by dismantling the classical non-self model and exploring the danger model, understanding exactly how the kinetic violence of trauma shatters cells and releases damps like HMGB1. We tracked those endogenous alarmins to the TLRs on the macrophages, watched the MyD88 cascade through the NF-kappa B switch, and witnessed the massive IL-6 cytokine storm. We detailed the physics of neutrophil rolling, the destructive power of the respiratory burst, and how the extrusion of nets directly links the innate immune system to the catastrophic massive coagulopathy and the destruction of the endothelial glycocalyx. We mapped the macroscopic clinical phenotypes from the hyperdynamic SIRS response through the dangerous window of the two-hit model, all the way to the immunosuppressive catabolic nightmare of PICS driven by MDSCs. And finally, we discussed the absolute biological necessity of targeted nutrition glutamine, arginine, and essential electron sinks to fuel the fight for resolution.
And for you, the practicing critical care surgeon, the practical applications of this deep microscopic dive are immediate and clear. First, your early, rapid, massive resuscitation utilizing balanced blood products in a 1.1.1 ratio is not merely about refilling an empty intravascular tank. You are executing a highly targeted biochemical resuscitation of the shattered endothelial glycocalyx, directly mitigating the profound generalized endotheliopathy that drives consumptive DIC. Second, your deliberate disciplined timing of major orthopedic and general surgical interventions, embracing the philosophy of damage control, is rooted entirely in preventing a massive exogenous damp release in a patient whose immune system is already massively primed. You're actively avoiding the lethal second hit that triggers fatal ADARTs. And finally, your daily ICU management of the complex chronically ill patient must proactively anticipate the dangerous transition into CARs and PCs. It demands aggressive, early, and highly targeted nutritional support, utilizing specific amino acids and antioxidant cofactors to fuel the enterocytes, protect the T cells, and mitigate the profound immunosuppression that leads to late sepsis recidivism.
Which leads me to a final provocative thought to leave you with, building directly on what we just discussed regarding the active resolution of inflammation. We spend so much of our time, our energy, and our pharmacology in the surgical ICU desperately trying to block inflammation. We utilize high-dose corticosteroids, we aggressively pursue surgical source control, we deploy continuous renal replacement therapy, and we use massive fluid resuscitation strategies, all in an attempt to blunt, suppress, or wash out the systemic fire. We are constantly trying to throw a wet blanket on the cytokine storm. But if the transition to actual healing, if the true resolution of MOS isn't just a passive fading of the fire, if it is, in fact, an active genetically commanded process driven by the targeted synthesis of molecules like resolvins, protectins, and lipoxins that actively order the tissue to clear debris and rebuild itself, are we approaching critical care resuscitation completely backward? In the future, instead of just using blunt, non-specific pharmacological tools to simply block the collateral damage, will the ultimate edge in surgical critical care come from synthesizing, titrating, and directly prescribing the exact molecular signals, infusing the resolvins themselves, to actively command the patient's body to transition from destruction to reconstruction?
It is a profound paradigm-shifting question. If we can harness the biochemistry of active resolution, we will fundamentally change our role at the bedside. We will move from simply being physiological firefighters desperately trying to limit the burn damage to becoming actual biological architects, actively commanding the tissue to rebuild.
And exploring those very questions is exactly why we do this. Thank you for joining us on this exhaustive deep dive into the microscopic chaos of trauma and the systemic inflammatory response. Keep pushing the boundaries of your clinical knowledge, keep questioning the established paradigms on your rounds, and we will see you next time.